Welcome to Group 7 presentation, design and expression of multi-epitope peptide antigen against Vibrio cholera. Cholera is a diarrheal disease characterized by watery stool, vomiting, and may end up in death. It is caused by the bacterium Vibrio cholerae. In Nigeria, there have been many outbreaks of cholera as depicted by the image to the left. The yellow dots show cholera detected, agents detected using a rapid diagnostic kit. And the red dots show the bacterium detected using culture method. Serotype 01, 0139 have been indicated in such outbreaks in Nigeria. Nigeria, amongst African countries, experience sporadic cholera outbreaks, and definitely significant cases reported and some deaths have been recorded. And this begs for the development of an effective cholera vaccine, which should mitigate the impact of such diseases. Hence, we're exploring a synthetic biology approach to develop a multi-epitope-based cholera vaccine. The pathogen, Vibrio cholerae, colonizes the intestinal gut and as such stimulates the cells there to use water, and this accounts for the watery stool. The enzyme, the bacterium produces a toxin, cholera toxin, which is composed of two subunits, CTXA and CTXB. And also, virulent factors produced by the bacterium is secreted outside, it's exported outside the cell via the type 2 secretory systems. On the outer membrane of the bacterium, we have some proteins, outer membrane. This pathogen releases virulent factors and it is transmitted by unhygienic conditions like contaminated water and stool. The immune system, the innate immune system, plays a crucial role to the initial response to Vibrio cholerae infection. The macrophages and other innate immune cells recognize pathogen as molecular patterns of the bacterium, and so they now release inflammatory cytokines. The adaptive immune system, the beta cells and T cells, produce cholera-specific antibodies, which target cholera toxin and the lipopolysaccharides. Vaccine development. Also, as of now in Nigeria, the oral vaccine administered whole-cell killed whole-cell bacterium or attenuated organisms. But recently, subunit vaccines have been made which are more effective than the whole-cell vaccines. But currently, epitope vaccines are the latest vaccines that are being produced. And one polyvalent vaccine has been produced by Rukayat et al. 2022. Hence, in our own setting, due to our local terrain, we propose to make a multi-epitope vaccine using our strains isolated from outbreaks here in Nigeria. And our solutions. Over to you.
Our solution involves the development of a multi-epitope-based cholera vaccine, which we have named the COT-COL epitope vaccine. How do we go about this? The steps involves identifying antigenic epitopes of the Vibrio cholera pathogen, which include the toxin itself, the outer membrane proteins, and the type 2 secretory cells. We would also carry out epitope linkage after identifying our epitopes of interest and codon optimize this based on our identified chassis. Would go on to carry out immunogenicity assessment, then the synthesis and cloning of our construct. Then would express our proteins in E. coli BL21 and isolate this protein for preclinical evaluation. Next slide. Yes. The very first step, which involves the epitope prediction, is a very critical step involved in multi-epitope based vaccine development. And why is this? Whatever design we produce, we have to make sure that it is capable of inducing humoral immune response. And how is this done? A series of bioinformatics analysis is carried out to ensure that we are able to produce an antigen that is capable of inducing innate and humoral immune response. So the steps involve the collection of data, the proteome and nucleotide data from appropriate database, B and T cell epitope prediction, antigenicity prediction, allergenicity prediction, as well as the MHC1 and MHC2 prediction. All these and more are important steps that are involved in identifying the most immunogenic epitopes that are capable of inducing immune response in the host cell. Once the series of epitopes have been identified, they would be put together to form our antigen. And this will be done by linking these epitopes together with specified linkers that are capable of linking the HTL epitopes and the CTL epitopes. The full epitopes are predicted for their secondary and the tertiary structure. Also, would also consider adding an adjuvant since scientifically it has been studied that protein-based epitopes require additional immune stimulation. Next slide. Here is an illustration of an antigen that was developed by Shea et al. In 2019. Here they have linked HTL epitopes and CTL epitopes together using specialized linkers, which include the EAAK linkers, the GPGPG linkers, and the AAY linkers. These linkers have been reported to be capable of linking epitopes together and also helping to maintain the immunogenicity of each epitopes at the end of the day. Next slide. Yes, these designed epitopes are codon optimized because we want to make sure that our host is able to synthesize the protein and maintain its conformation and also be able to induce the immune response that we need. Lastly, we would also need to carry out molecular docking assessment and the immune simulation. These are all steps that will help us validate our antigen that we have designed.
Plasmids with features such as strong promoter, antibiotic resistance markers, and origins of replications would be selected to ensure stable maintenance and propagation of plasmid. Cloning of the genes of interest will be done using the ligation-independent cloning strategy, which makes use of the 3' to 5' exonuclease activity to create a long overhang for enough stability to hold this plasmid together during transformation and replication process. Next slide. Chemical transformation or heat shock method will be done using the competent E. coli as the chassis, which has a cell cycle time of 20 to 40 minutes. And this has the time duration for gene expression or gene translated for 10 seconds, which is good enough for the plasmid to be replicated and to be enough for the production of the vaccine. E. coli, next slide, please. Protein expression and purification. we'll be using auto-induction. E. coli would be grown in specialized growth medium to automatically induce protein expression. Expressed proteins will be purified using affinity chromatography. Then, using SDS page, the molecular weight and the quality of the expressed protein will be known. Next.
The next part of our work will entail preclinical evaluation of the design multi-epitope vaccine and animal challenges. First of which, we are going to carry out immunogenicity assessments. This helps to measure the vaccine's ability to induce specific antigen-specific and T-cell responses. After that, we are also going to challenge some animal models we give them the protective dose of the vaccine, and then we expose them to lethal doses of Vibrio cholerae. And this old animal challenge model helps us to assess vaccines' ability to prevent and or reduce the severity of cholera symptoms. And then the last evaluation we're going to carry out here is the neutralization assay. The essence of carrying out this neutralization assay is to determine the ability of the antibodies that are elicited in the body to neutralize cholera toxins and prevent the attachment of the bacteria to the host cells. There are some advantages of this multi-epitope based vaccine over whole cell and subunit vaccine. The first advantage is that it has a pathogen-specific immunity is elicited, and this has been shown to be more effective in immune responses. It also ensures that undesirable immune responses are eliminated as a result of the specificity of the constructs. It generates prolonged immune responses. And then because we are using multiple epitopes, it improves the vaccine antigenicity and immunogenicity. And it can be developed, customized anywhere by persons with the required skills. Nevertheless, there are some disadvantages of this, one of which is that it can be quite challenging to find appropriate epitopes that can be combined in multi-epitope vaccine. And this also requires expert skills, which is inadequate in this part of the world. Also, the certainty of having proper and adequate attachment of the vaccine to the target cell is not always guaranteed. And then the chassis, which we are using to express the protein, sometimes might not express the protein correctly. In terms of safety measures, we are going to make sure that handling of the E. coli strains, especially the strains responsible for the outbreak in Nigeria, the 01 and 0339, would be carried out in accordance with different regulatory bodies, NIH and Ethical Review Board. We're also going to make sure that the activities and experiments relating to attenuated strains of E. coli will be carried out in biosafety level 1 containment. And the reason for this is because attenuated strains of E. coli are considered to be members of the risk group 1 agent. So it is very important that healthy humans are protected from them. Also, all personnel working with the strains would adhere to strict laboratory practices and use of personal protective measures. And tissue products and every other liquid generated during the experiments carried out in BSL level 1 and 2 will be disinfected and discarded in accordance with the required regulations. These are our references, and we are here to take your questions. Thank you.